This talk is a brief summary of how the cytokines signal and the different modalities that we have for modulating this particular signaling and dealing with pathology in different diseases. So in this cartoon, basically what I'm showing here is a brief summary on the more well characterized or better characterized TH profiles or phenotypes that have been associated with diseases. Starting with TH1, this particular phenotype is very well characterized as an inflammatory response. It is a response to danger, and basically it's the response we typically will have when we get a sunburn or a cut, or we are dealing with most pathogens, bacteria, etc. So this is a profile that's characterized by the phenotypic specialization of the T helper cells into the TH1, and it's going to cause a very inflammatory response. The TH2 is a response that's very characteristic of allergies, allergic responses, and this is the way that the body has to deal with different kinds of allergens, anywhere from food allergies to eczema to mosquito bites, and of course the diseases are very allergic. The Th17 profile is associated with the Th22 profile in humans, and basically the diseases have a component of inflammation, but they have been very well characterized. And it's typical, very actively typical for psoriasis, intestinal bowel disease or inflammatory bowel disease that comprises Crohn's disease and ulcerative colitis, and rheumatoid arthritis, psoriatic arthritis, ankylosing spondylitis. So these particular phenotypes are characterized for the TH response. And finally, we lead with the tolerance or immunosuppressive profile of the T regulatory cells, FOXP3. These are not linear. As I'm showing them here, they would be seemingly independent. But now we know that there's a lot of crosstalk and a lot of control and communication between these different phenotypic expressions of the different TH cells. And again, they can occur either in the normal situation dealing with danger, or they can be associated with very specific diseases. So the TH1, one of the products of that TH1 differentiation is interferon gamma, which was also one of the triggers that made the pluripotent T cell differentiate into TH1. So there's a bit of a retroactive loop that's positive, building up on that response. And this is something that we have all lived through when we fall and have a trauma, when we cut ourselves, or dealing with diseases. In a first stage, there's an increase. There has to be an augmentation of that particular cell that is perfect for dealing with that particular response. In a second stage, the products of these TH1 can also stimulate some of the cells that are characteristics of the TH2. In this case, particularly TNF has been shown to stimulate the B cells, which are very typically a response of the TH2. Now, interleukin-2 is inhibiting the differentiation to Th17, and it's a major product of the TH1. But it also inhibits the B cell, the isotype switch. So in other words, you have TNF stimulating the B cells, but IL-2 from the Th1 profile inhibits the switch. So the most characteristic B cell response in Th1 inflammatory responses is not going to be switched. It's going to be either an IgM, like when we have the rheumatoid factors, or it can go to IgG, but you don't see the full switch of the B cell a heavy chain to an IgE, which is typical of the Th2 response. So in this sense, there's a combination of crosstalk and there is a specialization into what is going to happen for that particular profile. The Th2 profile also has this sort of retroactive loop that's providing a positive feedback into producing more Th2s. And of course, with all the allergies, we do know that, you start sneezing, you start itching, and originally there's a very fast increase in that particular Th2 response. The Th2s will produce interleukin 4, which were also the cytokine that differentiated the pluripotent T cells into Th2s. But also some of the products of the Paneth cells and epithelial cells that are the distal end organ target of the Th2 response will be producing some cytokines that will stimulate further Th2 development. So there's this retroactive loop of growth. And there's also this crosstalk. The B cell is actually not only responding to interleukin 6, but it's also producing interleukin 6. And interleukin 6 can actually have signaling effects on the inflammatory pathways, not only, as you can see here in this cartoon, having effect on synoviocytes, fibroblasts, and even osteoclasts and cartilage. But if you go left in this cartoon, interleukin 6 also has effects on the differentiation of Th17s, Th22s, and Tregs. So the production of some of these interleukin 6 by the stimulated B cells can actually lead to also some stimulation of other pathways in this Th phenotypic differentiation. Likewise, as all the other pathways, Th17s will produce interleukin 21, which is one of the cytokines that actually produces differentiation from the pluripotent cell into a Th17 cell. And interleukin 17, which is the major product of the Th17 profile, will inhibit the differentiation into Th1. So you can see right now here that generally a differentiation into Th1 inhibits via IL-2 a differentiation to Th17, and Th17 differentiation via directly interleukin 17 inhibits the differentiation into Th1. So there's a little bit of a control mechanism here that would probably tell us that diseases will have either one or another phenotypic profile, which would be more typical of a particular disease stage or even individuals. So not all diseases are going to be the same during the disease progress and also in different patients. Interleukin 17 also inhibits directly NK cells, which are a major target of Th1 responses. Now, what we see then is that almost all of the products of all of the other differentiations, either peripherally from neutrophils or even from the first Th phenotypic responses, the production of TNF can come from many different places. Interleukin 6, as I mentioned, also can be produced in different stages and different phenotypic expressions, and interleukin 2, together with interleukin 27, will eventually lead to a tolerance or immunosuppressive profile typical of a T regulatory cell. So all of these is actually characterized, but the important factor for this particular image is to say, how do we know it's a Th1 or a Th17? It's the cytokine profile. So the cytokine profile not only drives the differentiation on the very left, the first column of interleukins listed in there are cytokines. Not only the cytokine, the specific cytokine profile, it will determine what is going to be the differentiation, either Th1, Th2, Th17, 22, or Treg, but also each one of these will be characterized by the secretion and production of a very specific cytokine profile that helps identify what is the response we are seeing. So that's why it is very important to understand cytokine pathways and Th phenotypic profiles in the concept of the diseases that they're associated with, but also how they deal with danger. Because when trying to modulate these, if we understand what they're there for, we have to remember the immune system is not there for to cause diseases. It's there to deal with danger. So if a particular disease has a predominant pathway, we need to also remember what is that pathway normally used for and what could be the potential adverse event profile expected if I modulate that pathway. How is it that cytokines signal and create this complex system of communicating between cells? So what I have put in here is a generic cell that has receptors. These receptors could be receptors to soluble ligands or cytokines, or they could be receptors for homing or co-stimulatory factors. So whatever the receptor is on the cell, these receptors have the characteristics of having three domains. The extracellular domain is the one that will be specifically, the anatomy of it is going to be specific for binding the corresponding ligand. The transmembrane domain is the anchor, is the one that allows this particular protein, the receptors, to not leave the cell or not go into the cell. But it's the anchor that allows having one domain outside, the extracellular domain. And then the intracellular domain, that is the one that will produce the signal. The intracellular domain is the one that is actually bringing the message into the cell of what it has to do in response to what happened outside. So the cytokine is knocking at the door, but it doesn't come into the house. It just brings a letter. It just brings a message into the cell, and then the cell interprets that message. So it's the extracellular domain, which I call it more the anatomical domain. Then there's the anchor domain, transmembrane, and the intracellular domain, which I call the functional domain. Now, the way that they work, when I put this in an augmentation figure, is basically we have the receptors on the surface. Like I said, they're anchored, so there's a piece that's outside extracellular, a piece that's inside. And inside the cytoplasm, we have different kinds of kinases, which are basically enzymes that are going to allow a particular chemical reaction to occur by bringing energy through the system. Basically, it's going to be phosphorus, but there's other kinases that and other ways, but basically it's phosphorus that will produce the energy to create that chemical reaction inside. And there's different kinds of secondary messengers. So what happens is that when the receptors are engaged by the corresponding soluble factors, what the reaction is that the kinases will be activated, basically, like I said, by phosphorylation, and they then in turn activates the secondary messengers, which are now internal in the cytoplasm. Eventually they get to the nucleus of the cell, and the ultimate thing is gene transcription. So if we look at the example that I had before, you have, for instance, the interleukin 23 that will be engaging the receptor, and it will be phosphorylating internally to produce a gene transcription. That's the production of interleukin 17. That's one example. So IL-23 is one of the Th17 cytokines, and what basically happens is the engagement through this receptor system will produce this T cell into starting to secrete interleukin 17 and actually differentiate into a Th17 cell. So we can actually use either small molecules or biologics, which are more popular for this particular interaction. We can use them to block that binding or the engagement with the receptor. And the way that we can block this is either by blocking the soluble protein or blocking the receptor. So there's different ways that this interaction can be tampered with. And the whole idea is that by not allowing that engagement, what we have now is lack of activation and no gene transcription. So the particular cytokine that we may be blocking is still circulating. Cells may still be producing it, but it cannot engage with cells, and so it's rendered futile. It doesn't actually have an effect on the cells because we've blocked it. And this is the way that many of these extracellular drugs have been developed and used successfully to treat different diseases. So an example of extracellular targeting is the list that I'm going to say is just examples, is by no means exhaustive. So we can block the co-stimulatory factors, and there's several examples of interfering with the co-stimulation that has been successful. Rituximab, that blocks the CD20 receptor on the B cells, abatacept, CD40 in T cells, trastuzumab with the HER2 in breast cancer cells, and then the checkpoint inhibitors that are very interesting. I'm going to be talking a bit more about them. And I have other videos on YouTube that explain a bit more about the checkpoint inhibitors for immuno-oncology. These inhibitors inhibit an inhibitory pathway. So they basically allow the T cells to be more active against cancer. And then the cell adhesion molecules that also can block this interaction between the homing receptors and the migration receptors on certain conditions. The blockade of cytokines is probably one of the most popular ones. When there's a particular cytokine that is known, we can have inhibition of these cytokines by producing either an antibody or a recombinant protein that binds that particular cytokine and makes that cytokine not active anymore, not able to bind the receptor. So, tons of examples of different modulators of the TNF, IL-6, interleukin 12 and 23, interleukin 17, and several examples of interleukin 23 coming. And I'm sure that there's many, many more. That this is just, like I said, an example of how do we use this system to block cytokines. We can also block circulating factors. I mean, are they cytokines or not? They could be called cytokines. Are they growth factors or not? So they are circulating factors, and of course, bevacizumab and denosumab are two very good examples of blocking these circulating factors. And in the circulation, they're not bound to the receptor, and they cannot produce the effect that they're supposed to be producing. And then we can also use this system to produce agonism. So another thing is to say, well, can I actually use an endogenous protein and actually give that to patients to promote that particular signaling in the cases where we can perceive there might be a lack of that. And examples are, for instance, the GCSF in neutropenia. We can stimulate the neutrophil growth, or epoetin for anemia. You can induce the growth of or the intracellular signaling by actually binding to the receptor in patients that may actually have less amount of that. So modulation of these interactions with the different cell receptors will allow us to understand if we are increasing or decreasing that intracellular signal, which is the end result that we are looking for. So when we actually look at the different Th phenotypes and how they have been associated with different diseases, we can understand what would be some of the approved biologics that target cytokines. So in this particular cartoon that I have created, and I used in several different educational talks, I just wanted to show some of the approved biologics and where they work. So when we look at abatacept, way on the left, so at the very initial co-stimulatory signals with the T cells, abatacept would be inhibiting that co-stimulation. And by inhibiting it, even though there might be antigen presentation by dendritic cells, the T cells are not responding because they lack that co-stimulatory signal. The checkpoint inhibitors, if you look at the example, for instance, using a CTLA inhibitor, also on the left, what you see here is that in this case, the checkpoint inhibitors, the CTLA, will be blocking that co-stimulatory signal, in this case, coming from the dendritic cell, the presentation cell. This is a very interesting area of research in immuno-oncology because this particular pathway inhibits the T cell from responding. So by blocking the inhibitory signal, the T cell will be just active. It doesn't have that particular control or the break that it would normally have. And this T cell would be then very active, which is the reason why it's used to treat cancer, because this way the same cell from the individual will actually be just constantly active and dealing with all of these tumor antigens, and the cells that actually carry that tumor antigen will be targeted by these T cells that no longer have an inhibitory pathway. They have been blocked. Another way to do this blockade is by using the other inhibitory pathway in the T cells, which are in the periphery. So in this case is the PD-1 or PD-L1 system in the peripheral T cells that have already been differentiated to identify the tumor antigen, and this is the binding to the cell that has that tumor antigen. So this is the T cells that bind the antigen in the cancer cell, and they are no longer inhibited, so they will be constantly active and killing those tumor cells because they have this PD-1, PD-L1 inhibitor. So the checkpoint inhibitors inhibit an inhibitory pathway and render the T cells more active. Rituximab, which is a CD20 antibody, it will block the binding of the it will block the receptor, the CD20 receptor on the B cells, and it basically inhibits the B cells that are maturing. So it will have an effect on the diseases that are very typically B cell related. Ustekinumab is going to be working basically on inhibiting two different cytokines. And this is quite interesting because the two different phenotypic pathways, Th1 and Th17, at least in vitro, they are seemingly opposite, and each one will actually inhibit the production of the other. The ustekinumab well, actually is an antibody that binds one of the chains that is common for both cytokines. So interleukin 12 and interleukin 23 have two chains, but one of them is called P40 and is exactly the same for these two cytokines. So ustekinumab will render IL-12 and IL-23 unable to engage with their receptors. So in a disease that's predominantly Th1 uh, uh, and may actually have already inhibition of Th17, ustekinumab is probably inhibiting more of the IL-12 pathway. Likewise, if there's a more predominant Th17 disease, ustekinumab will be binding more of the IL-23. But there's actually some advantage to understanding diseases and disease stages where maybe blocking these two differentiations could be good for complex clinical pictures or for diseases that may actually be having some sort of different profiles either at different stages or in different organs. Tocilizumab you see on the right, inhibiting IL-6. It inhibits the end product in the synoviocytes and the rankle stimulation, but it also will inhibit the differentiation into Th17s, 22s, and also the T regulatory cells. So interestingly, interleukin 6 will be a good inhibitor for the end products. So I put them like from left to right. Left is more central and the right is more peripheral effect. So what you see there is Th1 more peripheral effects of IL-6 are destroying the cartilage, the bone, and are producing joint inflammation. But going more to the left, IL-6 will also the inhibition of IL-6 will also be inhibiting the Th17, 22, and Treg. So IL-6 has a potential, in theory, of treating many different conditions and stages. Interleukin-1, as you can see, is a product of macrophages and very much associated with an end product in Th1 diseases, but it also seems to be involved in Th17 proliferation. However, the experience with IL-1 has been more successful for treating familial Mediterranean disease and other diseases that are identified as more purely IL-1, IL-18 pathway and the inflammasome pathway associated. Where you have diseases that have more cytokines involved, it does seem that inhibition of IL-1 may not be sufficient, which of course we have seen with TNF. There are many TNF inhibitors. There's many different ways. So we have recombinant proteins or antibodies to TNF. And TNF is a pluripotent cytokine. It is produced not only by many different cells, but in the many different compartments and at the very different stages. So TNF unquestionably has shown during decades of use a very successful pathway for inhibiting it in inflammatory diseases and is not just the ones that are more predominantly Th1. So, TNF is efficacious in typically Th1 diseases like rheumatoid arthritis and also IL-17 pathway diseases like psoriasis or IBD, inflammatory bowel disease. TNF does have a lot of different functions. And in this cartoon, you can see it in the many different stages as playing an important role. By the same token, you have something that's actually used by many different cells, and you need to make sure that you look at the potential adverse events of all of these potential inhibitions of pathways that are really there to help us deal with danger. Now, on a later stage of more specific cytokine targeting, I want to talk about the IL-17 inhibition, which is, as I mentioned, it's sort of a much more specific. It's only one cytokine. It seems to be very limited to the Th17 profile, but in the clinic, it has shown a dramatic efficacy for psoriasis, psoriatic arthritis. So diseases that are very much IL-17 specific. It's a bit the same as what happened with interleukin-1 inhibition and the inflammasome diseases. So the more specific cytokine profile the disease has, the best results we're going to have by inhibiting that particular specific cytokine. And that can happen also with the cell adhesion molecule inhibition in the case of vedolizumab, that is inhibiting the T cell migration to the pNET cells and epithelial cells in the guts. So it's very specific for inflammatory bowel disease, specifically, Crohn's disease, ulcerative colitis, and blocking this very specific migration and adhesion molecule that brings the T cells to the tissue, inhibits that migration, inhibits that activated T cell from entering and causing more inflammation, and has been very effective. So the different pathways that we understand characterize the different disease stages or diseases itself can be blocked by many different extracellular mechanisms. And what's very important is that when choosing one or another, we have an understanding of where is this working, why do I think this would be of the best benefit for a particular individual or patient, and do I have a different choice that could be either better or worse, or if what would be second best or third best, etc. So the pathways are important to understand so that you know exactly what to choose when you are selecting a treatment modality for patients. So the binding of cytokines or the engagement of homing or co-stimulatory factors will produce an internal signaling, and blocking that particular interaction extracellularly has been well received and well characterized using biologics. But there's another way that we can also block that signaling. So what happens when the cytokine or the co-stimulatory factor bind the receptor on the cell through the extracellular domain? This actually brings an activation basically by phosphorylating the kinases and the different secondary messengers inside the cytoplasm, and eventually produce gene transcription. So this could be just a brief example, could be IL-23 is binding the receptor, activates the kinases, and the gene transcription is producing interleukin-17. So this is just one example how interleukin-23 actually is differentiating that T cell into a Th17 cell that is secreting IL-17. So we can block that with external blockade by either small molecules or large biologics, or we can find the alternative of small molecules that can target these intracellular pathways. So in this case, the small molecules penetrate the membrane. It's not like the large biologics that they have to stay outside the cell. The small molecules penetrate through the cellular membrane into the cytoplasm, and they actually bind any one of these particular areas where normally the kinases would be phosphorylated. But by not being phosphorylated, by blocking the phosphorylation, note that the engagement of the extracellular ligand can still happen. But basically, what you have is a kinase that is not active. And by not being active, there is no gene transcription. So either blocking extracellularly and not allowing that binding to happen, or allowing the binding to happen but not allowing the activation intracellularly to happen, we have the same result of no gene transcription. So bottom line, that cell will not be responding. And that's another way of blocking these cellular crosstalk and communication. So there are several receptor superfamilies, and what I put in here was just reminding you that these cellular receptors are present in many cells. They're not unique to the immune system cells. They're present in many cells, and that's what allows cells to produce the cross-talking, the communication, and the responses. The receptor superfamilies are characterized by basically internally. So what I depicted here in blue is like the cytoplasm, and then you of course have the nucleus and the gene transcription in the bottom. So the first superfamily is a type 1-2 of cytokine receptors, and they are characterized by having tyrosine kinase association, and they signal via the STATs, the signal transduction and activators of transcription. So this is the way that the first family will be communicating to the gene. The second superfamily is the one we know as the toll-like receptors, which is the one that the interleukin-1 uses. And the intracellular signaling is a bit more complex. It involves MyD88, which is an adapter protein that will produce several intracellular messengers for the signal activation, which is TRAF and IRAC signals, and then they go to the NF-kappa B for gene transcription. So signaling internally, the first one is tyrosine kinase and STATs, and the second one includes TRAF, IRAC, and NF-kappa B. The third family is the family that's the NF-kappa B family and TNF. This is the one that tumor necrosis factor uses. And also the BLIS for the B-cell activation uses this kind of receptors. Basically, this is trimeric receptor, so there's 3 chains, and they will signal via the NF-kappa B and the MAP kinase. We have the TGF-beta receptor. They use serine or threonine kinase, as opposed to the first type 1-2 cytokine, which used tyrosine kinase. The TGF-beta receptors use serine or threonine. So that's what differentiates this superfamily from the others. The chemokine family will now use CAMP, so cyclic AMP. It's a G protein. It's not the very typical of the others. This is unique for the chemokine receptor, and they also be activating via the phosphodiesterase 4. So the PDE4 is the via of communication for intracellular signaling. And finally, the last of the 6 superfamilies is the receptor tyrosine kinase. So in this case, the enzyme-linked receptors. So this particular receptor does have the tyrosine residues within the chain, as opposed to the first one, which doesn't have a kinase in the receptor, but the kinase is associated physically. It's a different protein associated to the receptor in the type 1-2, whereas in the receptor tyrosine kinase, the tyrosine residues are in that receptor. And that's what characterizes the 6 different families of receptors. It is important to have an idea of what exactly is the mechanism for a particular communication or pathway in disease stages, so that we can understand what exactly are we modulating, where are we modulating, and what can be the expected results of this modulation with the different armamentarium of drugs that we have. So as an example, the different drugs that will be using or blocking in the different superfamilies we have for the type 1-2 cytokine. There's different examples. We have extracellular tocilizumab, ustekinumab, and intracellular litofitinib as examples. There's many other examples of these binding. Anakinra is the typical IL-1 inhibitor, so that's the one that would be using this particular pathway and inhibiting it. Of course, for the NF-kappa B and TNF, we have the many different TNF inhibitors. This is just an example of the first that were approved. And belimumab is another one because the BLIS also uses this particular receptor type. We have chemokine inhibition by fingolimod, which is an intracellular inhibitor of the cAMP activation, and PDE4 inhibitor. And we also have the receptor tyrosine kinase inhibitors of bevacizumab that will actually inhibit the binding of VEGF to its receptors. So there are several different ways of modulating and blocking, and there's also in development, TGF-beta is an area of development in immuno-oncology, both intra and extracellular. And they are, in development phase. We have no idea when anyone could be approved, but all 6 superfamilies are in one way or another being studied carefully and used for modulating them and treating diseases. So in this cartoon, what I'm showing is a hypothetical cell. I don't think there is any single cell that actually has all these receptors in the same cell, but I just wanted to show how it would look for all of you on the cross pathways that we have. So on the map signaling cascade on the very left, what you see is different secondary messengers that are being activated, phosphorylated to lead to gene transcription. And you can see some crosstalk with a sick pathway cascade that actually eventually goes to the JNK and the ERK. Phosphorylations and gene transcription occurs with a little bit of cross communication between the SYK and the MAP kinase pathways. The P13K cascade has several different messengers that are actually shared with the Toll-like receptors, and the Toll-like receptors also can cross-talk and communicate not only with the P13, but if you follow the arrow, it goes also to the kinases associated with the MAP kinase signaling cascade, and also to the NF-kappa B cascade. So as you can see, the cAMP or the chemokine cascade, it's a bit more directly going down in the mauve color, or sort of a magenta, bluish magenta color. It goes more directly without cross-talking. The PKC signaling does crosstalk with the NF-kappa B signaling cascade typical of TNF, which also jumps over to the left on MAP kinase. And also the Jak signaling, similar a little bit to the chemokine cascade in the magenta. Now the light turquoise color Jak signaling cascade is also more direct and has less crosstalk. Why is this important? Well, it's important to understand if you're using a particular blockade, you can block extracellularly or intracellularly. And there are some advantages to both. So if you are ever posing a question in your mind, what is better? Is it better to use an extracellular block, or is it better to block intracellular? Well, the answer is yes. In other words, it depends. You need to know which circumstances would extracellular be an advantage, and in which circumstances would intracellular be an advantage. But they are different, and it's important to understand why they're different. So in order to explain a little bit about why one would choose an extracellular inhibition versus intracellular inhibition, I want to discuss the NF-kappa B signal cascade of TNF mediation and the MAPK kinase signaling cascade, which crosstalk internally inside the cell. What you have here is a cartoon on the left that depicts more or less the interpretation I have drawn from reading the many different papers on all the different secondary mechanisms and messengers that are activated just by TNF binding the receptor, or the IL-1 binding the receptor. But let's talk about the TNF binding the receptor. As you can see, it's complicated. There's a lot of different kinases and mediators that are jumping around and passing the baton in this race. There's a lot of different things that are happening before we get an inflammatory response in the gene transcription. The cartoon on the right, I actually have taken it from the Wikipedia, and I thought it was actually extremely good for in one image showing the complexity that you have intracellularly. And this is without showing the communication with the toll-like, just the TNF. Look at how many proteins intracellularly are actually engaged in different pathways with TNF, and how you have different effects, including apoptosis. It's not for anything. I mean, it's actually not inconsequential that TNF is a tumor necrosis factor, because it does cause cell death, and very nicely depicted here and very clearly that you can cause cell death with tumor necrosis factor, basically on tumor cells. So that's one of the effects of the tumor necrosis factor. But as you can see, there's a lot of things that are happening inside the cell. If we want to inhibit the effect of TNF because it's producing or associating with a very inflammatory disease, and in this case we want to modulate and inhibit it, if we wanted to successfully inhibit the inflammatory response using a small molecule intracellularly, we could not choose just blocking the MKKK, or just blocking the TRAF2, or just blocking the JUNK, or just blocking the NF-kappa B, or the IRAK4. So if you choose only one intracellular blockade, the other systems, the other proteins could be activated. The redundancy of the intracellular signal to produce the effect when TNF is engaged in that cell is telling us something very important. It's telling us TNF is super important for us to be able to live and survive. TNF is used for most of the pathways when we deal with infections, when we deal with cancer, etc. So it is a very important cytokine. But when this cytokine is going crazy, we need to control it. and to control it intracellularly would not make much sense. Uh, the patient would have, you would have to develop maybe, 4 or 5 different drugs. Patient would have to take a lot. So in this case, just by blocking extracellularly the TNF, just by producing a biologic that can bind TNF and successfully inhibit it by blocking the binding of that TNF to the TNF receptor, you can control all of that intracellular signaling, because by not engaging the receptor, there will be no signal. So in the case of signaling that is very redundant intracellularly and where there's too many intracellular pathways, it is a better way to block extracellularly rather than trying to find intracellular blockade. In this particular cartoon, again, this is a very complex and hypothetical cell that is showing all the different crosstalk of the different receptors, but in the Jak system, the Jak Signaling Cascade, it's a very direct cascade via the receptor, the Janus kinases, and the STATs, the signal transduction and activators of signal translation and activator of transduction. So the way that the Jak or Janus kinase system works is very interesting, and there's a beauty in the simplicity of it. There are 4 Jak family members. It's a Jak1, Jak2, Jak3, and TYK2. There are 7 signal transduction and activators of translation, which are STAT1, STAT2, STAT3, STAT4, STAT5A, STAT5B, and STAT6. The Janus kinases are associated with the internal portion of the receptors. So these receptors, which are the cytokine type 1-2 receptors, in the intracellular tail will have 2 Janus kinases associated with them. And these 2 Janus kinases that associate with them will depend on the receptor. As you can see in this cartoon, we have different cytokines, and this is just an example. So the gamma chain cytokines are the cytokines associated with the white blood cell lineage. And then you have the interferon that will bind a receptor that has a different pair of JAKs associated. Interleukin 10 and 22. Interleukin 23 and 12 have TYK2 and JAK2, and so on. So different cytokines will have slightly different specialization for the cytokine that they're binding, and intracellularly the pair of JAKs that they have. Just note that the IL-6 and IL-11 column, the intracellular Janus kinases are 2 of them. But for this particular group of cytokines, any combination of these 3, so it could be Jak1 and TYK2, Jak1 and Jak2, Jak2 and TYK2, any one combination of a pair with these 3 kinases will be associated with the receptors of the cytokines, IL-6, IL-11 families, that will signal through the STAT 1, 3, and 5 for transcription. The only example that I know of, and to date I don't think there's been a publication on any other example, where 2 of the same Janus kinases are associated in a receptor, is a typical example for Epub and for the CSF growth factors. These are not cytokines, these are growth factors. And that's very important for the cell to differentiate which kind of stats and which kind of signal you're going to give to the cell. In the other signaling, what you could be signaling is a synthesis of a different cytokine. In the EPO and the growth factors, what you are signaling to the cell is basically replication of that particular lineage cell. And this is a way they have to communicate. So it's very important to understand that the 4 Jak family members have different combinatorial abilities, and also via 7 different stat members on different cells, we can have a tremendous amount of different kinds of responses. So how is it that only 4 Janus kinases and only 7 signal transduction and activators of translation can actually produce so many different results? Well, I'm no mathematician. There's a mathematical field on combinatorial power and the power of combinations. This is just a schematic that can actually show how having 4 elements, you can have how many different combinations, and of course then you have rules of combinations. You can say, X does not combine with Z, but X can combine with Y, whatever it is. So these combinations actually create a much richer amount of data than if we had to have each one as a whole. So power combinations is a complex field of mathematics that we actually use every single day of our lives, because we all use 26 letters that have the power of being combined into words. And just a few, a little question for you if you're listening and paying attention and liking this. Just have a wild guess. How many words can you create with 26 letters? And of course, there are some rules of combinations. Sometimes the rules can be X letter cannot go before the other one, or you cannot end a word with a particular letter, but in general, you actually have a power of combination that is tremendous. So if you guessed around, I don't know, 200,000, you probably were on the right track. But that's only if we're counting English. These letters can be combined to form words in many, many languages. So the power of combination is tremendous, and in the cell, having the minimum amount of elements allows the cell to have less DNA committed to having a message. So if we had to have one gene for each one of the proteins, or one amino acid, one nucleic acid for each amino acid or for each protein, I think we could not be an average weight of 60 kilograms or 120 pounds. We would probably be 1,000 times more heavy, or even more. We could not contain all that amount of DNA in our cells. So we need to be economical in the cell and make sure that we have the minimum amount of elements with the maximum combinatorial power, so that all of these different combinations give us all the different responses that we need to have in the case of the immune system to deal with all of the different perceptions of danger that can be from pathogens to sunburns or cuts, and also they're used, unfortunately, they can produce diseases. But this is the important thing about combination and about signaling of cytokines that, for instance, are using the Janus kinase system with only 4 Janus kinases in combination. So when we look at the 4 different JAKs and the different receptors that have been described, we have the JAK1, JAK3 receptor, which is the receptor called for the gamma chain cytokines, the JAK1, JAK2 receptors associated with that pair of JAKs. We have the receptor associated for the TYK2, JAK1, the TYK2, JAK2, and then the 2 combinations of any one of these 3. TYK2, JAK2, JAK2, JAK1, JAK1, TYK2, etc. So these combinations also in association with different intracellular signaling of the stats is going to be very important for the signal. So the gamma chain cytokines are basically associated with the white blood cell lineage, and they will be present in basically the cells of the immune system. The JAK1, JAK2 are more commonly used by interferons and basically produce a STAT1. This pathway is a pathway that deals a lot with viral diseases. The interleukin 10 and interleukin 22 are going to use different pairs of JAKs and different stats, the 1, 3, and 5. And interleukin 10 is a critical cytokine for regulation and immunosuppression or tolerance. So using the IL-10 in a very specific way with a specific stats, it depends on the cells that are expressing it, depends on the cells that are producing cytokine, and it depends on the stats that are going to be activated, what response we have. Is it an IL-22 response or is it an IL-10 response? In the case of IL-23 and IL-12, it's also very important to know which is the cell that is actually responding. IL-12 will be basically signaling through the TYK2, JAK2 and activation of the appropriate stats to produce a Th1 phenotype, whereas the IL-23, using the same TYK2, JAK2 and different stat pathways in different cells, will be producing or differentiating that Th phenotype into a Th17. So we have the same receptor, the same intracellular JAK association with different stats in different cells, 2 very different phenotypic expressions of the Th, either Th1 phenotype, more inflammatory, or the Th17 phenotype. And then in the case of the IL-6 and interleukin 11 and others, we have more combinatorial power, and we have also different stats intracellularly. So these combinations allow for a lot of different responses. IL-6 is actually present and it's activating, and it will bind many different kinds of cells. It will produce many different kinds of responses, basically many of them inflammatory, and it will also be involved in triggering IL-10, which is a regulatory tolerance mechanism. So IL-6 has a lot of different effects in a lot of different cells, and all of these different ways of communicating with the cells has to do with this combinatorial power of the JAKs and the STATs. And finally, the growth factors. So epoetin, thrombopoietin, and the granulocyte colonies stimulating factors, they basically will be signaling through the only known, at least to me, I don't think there's been another identical pair of JAKs identified for other signaling, but at least the pair of 2 identical JAK2 associated to these receptors are unique for growth factors. These are not for cytokines. And the stimulation via the STAT3 and 5 of these particular receptors and this association will produce cell growth. So these are growth factors for the epoetin, thrombopoietin, and neutrophils. So when we have cytokine signaling via the JAK family, that it is simple internally, but in this case, it's a very complex externally. So let me explain why I say that. We have the 4 different JAKs in this table, in the columns. But what you can see here is that internally, we can have different pairs of combinations of the JAKs that signal through particular stats. And that's what you see there. Stat 5 uses IL-7 for JAK3 and JAK1, IL-9, IL-15, IL-2, IL-4, and IL-21. These are the gamma cytokines, and they are present in the white blood cell lineage, and they are associated with an adaptive immunity response as shown on the top right. So JAK1, JAK3, many different cytokines. Intracellularly, more simple. Since these two have to be associated and active, just inhibiting one of them intracellularly can inhibit the signaling of all these kinases with only one single intracellular inhibitor. If we move on to the JAK2 TYK2, you see the IL-23 and the IL-12 will actually be binding a receptor that signals through the JAK2 TYK2. So again, these particular cells will be responding to the different cytokines, the two different cytokines, and you can inhibit that by having only one of the two JAKs inhibited. Now, if you go down the list, for instance, and you see, what if I were to inhibit only JAK1? You look down the list and you see all of the cytokines going all the way that have not only the adaptive immunity, but inflammatory T-cell differentiation, innate immunity. They all would inhibit a lot of different cytokines. If we had to inhibit these externally, extracellularly, we would need to have, probably an antibody for maybe each one of them. Maybe some of them can share some elements. But in general, to inhibit all of these cytokines, you can do only with one single targeted intracellular kinase, like the JAK1. That's one example. If you inhibited the JAK2 only, you are going to also hit the growth and hematopoiesis, the signaling for the growth factors. So in general, for treating inflammatory diseases, ideally you want something that does not inhibit JAK2 or that inhibits JAK2 at different concentrations or has lower affinity. And at the dose that you will give it to your patients, you're expecting it to block the other JAKs and not the JAK2. So in this cartoon is just a reference for saying intracellular inhibition by using the system, the cellular system of the power of combinations that has less amount of individual proteins working to produce many different signals. You can target one of those more simple intracellular mechanisms and block many different cytokine signals that could be involved in disease. In summary, for this particular talk on cytokines and their extracellular or intracellular blockade, do we need to know what we're doing? Should we know? I think we should. I think at least you should know not only the difference between large, protein or a pill. I think that when you explain this to your patients, it's actually good to give them the characteristics of the presentation. So you swallow it or you get it injected. But I think you should know a bit more about what are you doing inside the cell, outside the cell, in the organism. And is one better than the other? Well, I think you also need to understand a little bit about the immune responses and the inflammatory pathways. What are the immune responses in chronic diseases? Do you understand what is the innate and adaptive immune system that is contributing to this chronic inflammation. What is a cytokine-mediated TH differentiation? And I should also say, and what is a cytokine path or cytokine profile that characterizes that TH differentiation? So you can start understanding where with a particular disease or particular patient, like I say, sometimes it's not that one disease has necessarily always the same TH profile. It could change over time. So what is it that you're seeing from your patient that can help you identify what's wrong? Remember how they transmit the message from cell to cell. And then understand, when is the extracellular blockade better? Well, when you have a system of a lot of internal redundancy, a lot of internal messaging, the extracellular blockade is going to be more effective. So blocking only one particular cytokine, you can get a much better result of turning off all that cascade that's an inflammatory cascade. Now, intracellular blockade works excellent also when you have the opposite. You have many different cytokines that are using basically the same system intracellularly. So depending on the superfamily of the receptors that you're looking at, in the case of the Jak pathways, for instance, or the chemokine pathways, an intracellular blockade can actually help you better block a lot of combinatorial abilities that the cell has to respond to many different cytokines in many different inflammatory results by just blocking intracellularly a very critical path and a very critical drug. So you just need to understand what are you doing when you choose one or the other potential drugs, understand how to use them, understand the profile, understand the patient, but also know what exactly is happening in your patients when you are prescribing different modalities. Now, the other thing that might be good to understand is what are the drugs that selectively inhibit cytokine signaling? And, this is the important thing. If you have a particular cytokine that's super active in many different places, even though it is selectively blocking that particular cytokine, you know that you're affecting many, many different cellular pathways and many potential different effects. On the other hand, you might say, well, it's much more simple, just do a single intracellular message block. Well, yeah, intracellularly, like the Jak inhibition, could be perceived as single and very selective, but the reality is that these pathways are used by many, many different cytokines. So it's not selective for the cytokines that are going to use those pathways. It's selective only for the intracellular pathway that you're using. So important thing, to create this particular file and the animations and the cartoons, I have consulted many different literature reviews and different papers, but these are the ones that I would highly recommend, and I will put them in the YouTube description. Thank you for listening.